Hi, everyone, and welcome to the second UWECE Colloquium talk of the winter quarter. Today, I'd like to welcome Dr. Amy Orsborn, who joined our department in the fall of this past year as Claire Booth-Luce Assistant Professor in Electrical and Computer Engineering and also in Bioengineering. She completed her PhD in Bioengineering at UC Berkeley, UCSF, and did a postdoc at NYU's Center for Neuroscience, where she's been developing neural implants for investigating neural circuits in non-human primates. And she's very active here at UW in the Center for Neurotechnology. Her work has been supported by the NSF Graduate Research Fellowship, a predoctoral award from the American Heart Association, and also L'Oreal USA for Women in Science postdoctoral award. So please welcome Amy.
Great. Thank you very much. I'm very happy to be here at the University of Washington. Tell you a little bit about some of the work that I've done previously and how it kind of builds into themes that I'm going to be exploring in my lab here. So I'm really interested in brain-machine interfaces. The basic idea is actually really simple, is that we can directly interface the central nervous system with the external world. So for instance, you can make measurements from the brain, get information out to control something in the world. You can also take information from the world and put it back into the body with neural stimulation, like optogenetics or electrical stimulation. And this opens up a lot of new ways to think about treating neurological disorders. For example, if you have someone who's paralyzed because of a spinal cord injury, they still have intact parts of their brain that you could use to control something like a computer cursor or a robot to allow them to move again. And so there's a lot of different variations of brain-machine interfaces, all of them very interesting. I particularly study this application, trying to restore motor function to people with neurological disorders. And this idea has been around actually for quite a while, and there's recently been some exciting clinical trials showing that paralyzed people can actually regain function to be able to control something like a robotic arm to reach and grab objects. But there's still a lot of challenges if we want to take this therapy into a real-world practical therapy. Some examples are that the performance is really far from what we can do with our hands. Those patients wouldn't be able to do something like play a piano that requires incredible dexterity and high dimensional control of your hands. The devices also are relatively poor performing over long periods of time with a lot of variability. And there's also a lot of variability even across subjects. If you talk to someone in the non-invasive community of BCI, they might tell you about a term called BCI illiteracy. There's a subset of the population that actually can never figure out how to use an EEG signal to control an external device. And it's not really clear why that is. And so these are just some tangible examples, but sort of the bigger picture challenge is that we don't have principled or mechanistic understanding of how these devices work. And so from an engineering perspective, that means we don't have design principles. If you give me a particular patient with a certain injury, I don't know how to design the best interface for that particular patient. And so one of the reasons those design principles have been so elusive is because brain-machine interfaces are actually a lot more complicated than we originally thought. The initial idea was really that they were sort of feed-forward readout systems. And all we have to do is decode someone's thoughts. If I know exactly how your brain fires when you try to move your hand, I can build the perfect decoding algorithm to send those control commands to the robot and make it move like your arm does. But brain-machine interfaces are not feed-forward systems. They're actually closed-loop systems. So you have neural signals that are decoded to move the robot, for instance, but then the subject actually sees the robot moving and can change how their activity in their brain to move the robot in a different way. And so this is a closed-loop system that allows adaptation within the brain. And there's now decades of research showing that this is true, that you actually see neural plasticity and learning in brain-machine interfaces, including some really pioneering early work from Eb Fetz here at University of Washington and several other researchers here at UW. And so the sort of motivating principle of all of my work is that this learning and neuroplasticity can actually be an advantage, and that we can use that to improve brain-machine interfaces, and that we have to understand that learning if we're ever going to get towards this idea of having design principles to make brain-machine interfaces. And so we approach this with two sort of similar themes in the lab. The first you can think of as re-engineering brain-machine interfaces, where with this new understanding that they are adaptive closed-loop systems, how do we want to redesign them to optimize learning? So basically, we can treat the brain as some black box that we know is adapting in some way, but let's think about how we want to design all the rest of the interface to optimize learning within that black box. The other is that we actually want to study learning in brain-machine interfaces and get into the deep neural mechanisms. So we want to actually open up that black box and understand what might be going on in the brain. And the goal, of course, is that these two research arms will kind of communicate with each other, that as we understand more about learning, that will inform how we design the interfaces, and vice versa. Challenges as we try to develop brain-machine interfaces and tweak these different parameters might give us some clues about what might be happening in learning. And then finally, there's an additional thread of kind of looking at developing technology to try to improve these interfaces, and in particular, trying to actually improve our ability to study learning in brain-machine interfaces, which is sort of a whole brain phenomenon and happens in networks spread across the brain. So today I'm going to mostly focus on this first thread of research, thinking about how we want to redesign brain-machine interfaces given that the subject is learning. All right, so there's a lot of different ways that we can think about doing that, and this sort of serves as an outline for the talk. So first I'll talk a little bit about adaptive decoding, and then we'll think about how adaptive decoders will actually interface with the brain that's learning at the same time, so thinking about co-adaptation. We also want to think about how we design the rest of the loop, the sort of plant properties, and how that's going to influence control. And then finally, I'll touch briefly on some newer work thinking about how revisiting this question of how we pick the neural signals we use to control brain-machine interfaces. All right, so first let's start with adaptive decoding. So all the work that I'm going to tell you about uses sort of a common motor brain-machine interface paradigm for research. And so we have rhesus macaque monkeys that are our animal model, and we do chronic multi-electrode array implants in the primary motor and premotor cortex. So we put in electrodes into the brain, and then we can record action potentials of individual neurons. We use the firing rates of those neurons as inputs into decoding algorithms, and these are generally really simple linear filters, like a Kalman filter, and we use that to get out a control signal that's going to be a position and velocity of a cursor moving around in 2 dimensions on a screen. And the monkey can actually see that cursor moving on the screen, and so that visual feedback closes the control loop. All right, so given that an open loop decoder doesn't necessarily translate to this closed loop context, one strategy is to train your decoder in closed loop. So I call this closed loop decoder adaptation, or CLDA. And the idea is really quite simple, that when someone is using a decoder to control a brain-machine interface, you can look at the mistakes that they make and try to use that to update the parameters of your algorithm to improve the performance and correct mistakes. So this has been done by quite a variety of different groups, including some of my work. But I particularly was really focused on how to make these algorithms really robust and reliable so that you can learn a strategy that a subject is using in closed loop for control, regardless of where you initialize your decoder. And this is particularly interesting because you have to consider that the subject might also be learning. And so the rate at which you do this decoder adaptation actually matters because you can't assume that you have some stationary output signal from the brain. So one initial strategy that we found to be quite successful we call SmoothBatch. And the name kind of says it all in terms of what we're actually doing. But basically we have batch updates with short, small batches that allows us to get a quick update of our decoder parameters. But because this is relatively noisy, I want to then smooth them out and iteratively update over time. And so what this allows us to do is relatively quickly adapt the parameters of the decoder to avoid the decoder kind of chasing the subject if the subject is learning. So we want the decoder to adapt faster than the subject. And so the training signals that we're using, again, this is a supervised algorithm. So if you have a subject using a brain-machine interface and his target here is in orange, the cursor is here in orange, and he's trying to get to this blue target, and his cursor is moving off in the wrong direction, I can assume that he probably meant to go to my target here. And so then you use the neural activity in his brain plus what you think he's trying to do, what you infer, to get those new parameter estimates, and then you iteratively improve performance over time. So I'll quickly show you what it looks like when we do this. So here I'm showing you the success rate as a function of time, and in black is success rate, and blue is sort of an example of an error that he might make. So we start out with a completely random decoder, and he can't actually make mistakes or correctly perform the task. The cursor is actually sitting off in the corner of the screen because the parameters are just totally wrong. So gradually, as we start adapting, you can see that now he's actually able to make some mistakes as he moves the cursor around on the screen. He starts occasionally getting some right, and then he sort of the performance quickly improves, and then performance plateaus. And in fact, if we stop adapting here, performance remains high, suggesting that we've converged to some stable solution of a decoder that works well. And this algorithm was quite robust, so we tested it over 56 different sessions with a variety of different ways of having that initial guess of what the decoder parameters could be, none of them related to actual movement information in the motor cortex. And so in all cases, we started out with a very low performance, but we always got to a similar final performance. And we also were able to do this relatively quickly, getting up to maximum performance in about 20 minutes or so. So from here, we were sort of good engineers. We had some strategy that seemed reasonable, but we wanted to further improve this. So we saw that the decoder needed to adapt relatively quickly, and so we wanted to actually improve that more by adapting now the parameters at every millisecond rather than having these intermediate batches. And so we did that in collaboration with Miriam Shinichi. We worked on a point process filter decoder model where we could do this adaptation on a millisecond time scale. And so this improved our convergence significantly, getting down to good performance on the order of 6 minutes. So starting with a decoder the subject couldn't use at all to good performance in just a matter of few minutes. And we also tweaked this way that we were inferring what the subject tried to do, incorporating an optimal feedback control model to try to estimate other aspects rather than just re-aiming the cursor, also thinking about the speed that he wanted to control. And so this allowed us to actually get better performance in the final learned decoders. And so the subject was able to move the cursor faster without sacrificing any accuracy. So to quickly summarize this, we find that as long as you sort of consider the time scales, you can use decoder adaptation to learn a subject's strategy in closed loop. And we can do this relatively quickly and robustly, regardless of how you initialize your decoder. But one of the interesting things is thinking about how you would want to continue this strategy over days, right? So if this is an initial calibration, but how would we approach this if you come back the next day with this particular user? So to think about that, we're now going to move to these sort of questions of co-adaptation, or how do we want this learning decoder to interface with the brain. So one of the interesting things is that the learner, in this case the brain, is actually learning across multiple different timescales. If you think about learning to play piano, you get better within a particular practice session, but you also get better over weeks and months and years of practice. So learning has slow and fast timescales. And so if you start thinking about, issues of day-to-day variability in brain-machine interfaces, one of the challenges is that neural recordings shift from day to day. The particular neurons and are able to record, can move around, and so you have measurement noise. So one strategy would be just simply to constantly be retraining, right? So always do this recalibration phase every day. But when you do that, you can pretty reliably get to high performance across days, but there's actually quite a bit of variability here. And there's also no longer-term improvement. So this suggested to us that we're basically giving this particular user a new piano that he has to learn to play every day that has slightly different rules. And so that means that he's not actually able to get better and better over longer periods of time. So we wanted to think about how we might approach this to facilitate long-term learning in brain-machine interfaces. So we came up with one strategy for co-adaptation that's really quite simplistic. You initialize your decoder, you then do some decoder adaptation to get some reasonable decoder that the animal can control, but then you fix it after that. And the idea is to allow plasticity. So you want to keep the decoder the same for as long as possible so that they can learn it. If you have some sort of shift in your recording or measurement noise and performance drops, you then go back and tweak the parameters just a little bit. And so this allows you to sort of retain, the hope is that it would help you retain performance and gradually track shifts in the ensemble over time without disrupting that learning. And so I'm going to show you a video of what it looks like when you do this. So here is the performance on day one. This is just the initial decoder. Again, we've used no information from movement, so the cursor, he's really struggling to even get it to this central target in green. Now, after we've done this smooth batch adaptation to learn some of his control strategy. Now you can see that the performance is better, and he's actually able to go from this central target down to one of the peripheral targets. But there's still quite a bit of variability, and obviously that's not nearly as good as you can do with natural control. So now this is day 3, where we've left the decoder completely the same. We've just allowed him to learn and practice over time. And you can see that the control is getting faster and slightly more accurate, but of course there's still some kind of wiggles and jiggles in there. And then now if we zoom forward about 2 weeks, where we've done this sort of gradual iterative changing of the decoder only as needed, we can see that performance is actually quite good. And this actually looks very similar to what they would do in this task with their natural arms. All right, so just to quantify and show you what this looks like in terms of performance, this is the % correct on this center out task across different days. And so you start out very low performance, you adapt the decoder to improve it, and then allow them to practice, and they improve gradually over time, getting very close to the performance that they would achieve with their, if they were doing this task with their arm. And this worked in a second animal. And one of the interesting things we found is that this idea of taking this extra effort to sort of facilitate long-term learning might actually give us better performance than if we were to ignore the brain and just purely rely on machine learning. So what we did was really try to, on the first day, completely saturate performance. So we gave the animal a cursor, a decoder that he could control really, really well. He's at basically 100% on day 1. And he maintains that high level of accuracy over time, but if you look at the success rate, which reflects sort of how quickly and accurately he's controlling the cursor, you can see that there's clear learning and improvement. And so this suggests that, taking neural plasticity into account might actually give us better performance rather than just purely relying on machine learning. And then also just really quickly, the interesting thing about brain-machine interfaces is we can now look at what's happening in the brain as this learning is happening. And so one of the things we saw is basically that he's refining the way that he recruits neural activity to control the cursor. So here what I'm showing you is what we call direction tuning. This is the firing rate of a neuron as a function of the direction he's trying to reach. And so on day one, we have this sort of small cosine tuning relationship, which is typical of neurons in motor cortex. As he practices and learns, though, this neuron continues to have the same preferred direction. It fires most for this particular movement, about 270 degrees, but the amount that this neuron is firing increases. So he's figuring out which neurons are controlling the cursor and making them fire more. And this was pretty common. We in fact saw neurons that didn't have any relationship to the task start to participate over time. We also saw other changes in the temporal recruitment that I'm not going to go into, and these patterns actually stabilize over time, which is a hallmark of skill learning. So we think that he's sort of learning a skill of controlling this brain-machine interface that has a lot of potential advantages for long-term performance. So to sort of wrap up this section, neural and decoder adaptation potentially can actually interact synergistically, again, if we take into account some of these issues of timescales and the fact that learning happens across multiple timescales. And I didn't really go into it for the purposes of this talk, but we also see some interesting evidence suggesting that closed-loop decoder adaptation can sort of shape the neural strategies that the users find, which is potentially very interesting for sort of teaching people how to use these interfaces. This also gives us a lot of practical benefits where we can quickly improve performance and give someone a device that works on day one, but they can continue to improve on their own from there and get robust, reliable performance that we can maintain for long periods of time. And so one interesting thing that we're thinking about for future directions is understanding how this approach might scale up to higher dimensions. So all of this was done with two-dimensional cursors moving around on computer screens, but there's a lot of interesting questions of both how learning happens in high-dimensional spaces, like moving our 27 degrees of freedom of our hand, and then also sort of thinking about the right adaptive algorithms in these high-dimensional spaces. And this, of course, also requires a certain amount of technology development where we have to think about how are we going to study these movements in high dimensions. All right, so now let's move on a little bit to another project where we think about how assuming that sort of the brain and decoder are fixed in some stable way, let's think about how we design the rest of the loop, so the system that he's actually controlling. So just like the way that the biomechanics of your arm are going to influence how your brain has decided to control your movements, the properties of the control loop in brain-machine interfaces is also going to influence control and performance. So there's a lot of different, we decided to, in a collaboration with Miriam Shinichi, Helene Morman, and Suraj Gowda, we decided to look at, in particular, the rates that are in a brain-machine interface and how they shape control. So there's a lot of different rates, and I'm going to keep this diagram around for all of these slides because it can be a little tricky to keep everything straight. But basically, you have the rate at which you are estimating the firing rates of neurons, which is set by the binning of how long you're counting spikes for. You then have, and that determines the rate that you can actually decode a particular position. But then there's the rate at which a decoded position and velocity actually is sent to the cursor, which is a control, you can think of as a control rate. And then finally, there's the rate at which you actually show the subject that particular movement, which would be a feedback rate. So how often we're moving the cursor on the screen. And so we wanted to study the effect of the feedback and control rates, and this required that we had a decoder that was high rate and independent of, that operated in a rate independent way. And so the way we did this was with a point process filter that decodes activity on a spike by spike basis. So rather than something like a Kalman filter where typically you're sort of just operating on binned firing rates and assuming Gaussian properties of the neurons, here we're modeling each neuron as a Poisson process, and that allows us to decode on a spike by spike basis. And so now we can decode at a millisecond time scale very quickly and then manipulate these other two rates to see how they influence performance. So the manipulations that we did are, you can think about, one would be having a what we call a fast decoder, where we're decoding very quickly every millisecond, and then we're controlling the cursor every millisecond, and also pushing those updates to the screen at every millisecond. And so this is both fast control rates and fast feedback rate. Alternatively, we could actually sort of downsample the control and feedback. So again, now we're decoding at every millisecond, but we're not pushing the decoded velocities to the actual movement of the cursor until a lag. And similarly, we're not pushing the update to the screen. And so here you've got a slow control and feedback rate. And then finally, you can actually also do a manipulation where you have fast control rates but slow feedback rates. So you're still controlling the cursor at a very high rate, but you're not showing it to the subject. All right, so with those three combinations, we can now actually compare to isolate the effect of feedback and control rate on performance. And so the first is comparing a slow feedback, slow control rate with a fast control rate, slow feedback rate decoder. And so this is isolating the control rate. So what happens when we actually increase the control rate. And so this is the success rate. And so higher is better here, and then this is the delta in success rate between these two bars here. So what you can see is that by increasing the control rate, we gained about not quite 10% fold improvement in the performance. And this is actually very interesting because this is faster control, but he's not actually seeing it, right? So the cursor is moving quickly, but he's not actually getting feedback about the time scale that he's controlling the cursor, but it still improves performance. Next, we wanted to look at the influence of the feedback rate. So what happens when we go from fast-slow to fast-fast. And so here you can see that again, by increasing the feedback rate, we actually gain another not quite 10% fold improvement in performance. And then finally, we wanted to see whether these two effects were separate from each other. And so if you compare the slow-slow to the fast-fast, we now see a larger improvement that's basically the sum of those two performance improvements. And so together, when you have fast feedback and fast control rates, you can get significantly higher performance in these systems. Yeah?
Just for some context, you mentioned that fast is about 1 millisecond per sample. What's slow in this instance?
Yeah, so that's a great question. It's with 100 millisecond bins, yeah.
So it's slower than the visual feedback rate, like 60 hertz.
Right, yeah.
Gotcha.
Yes, great question. There's some other interesting aspects of that project, but for the purposes of this talk, I'll sort of keep it short. And so this project, I think, is sort of potentially interesting because it really highlights the fact that feedback and the properties of your control loop obviously are going to influence performance, and so we actually really need to, and it also actually, this sort of isolation of feedback versus control rates suggests that there might actually be both feed-forward and feedback control strategies that are being employed by the user in this interface. And so obviously this also highlights that we really need to fully design this entire closed loop system. We don't want to, we have to think about, how we choose to decode is going to set a certain rate, which will then influence and have propagating effects with respect to control. So if we purely think about maximizing decoded information, we might be sort of short-sighted and shortchanging ourselves because the rate, for instance, matters. And then I think this also really highlights the fact that the more we can understand about kind of the learning strategies of the user, the better we can optimize the system. So again, going towards the fact that we need to have proper models so that we can have design principles for these interfaces. And right now, we know that this is learning and we can have some inferences, but we don't really know very many details about how this learning or control is happening. All right, so in the last little bit, I want to talk about some newer work where we start to think about how we select signals for brain-machine interfaces. So as I've talked about previously, we always thought for a long time about brain-machine interfaces as decoding the brain. And so that informs how we think about what neurons we want to measure and how to control these brain-machine interfaces. So there's a lot of actually different measurements that you can make from the brain. Just thinking about electrophysiology, we can put in electrodes into the brain to measure the action potentials of individual neurons, called spikes. And this is what all of the research that I've talked about so far has used action potentials. But with this penetrating electrode, we also can get a lower frequency signal that we call the local field potential that basically is thought to reflect kind of summed activity of a bunch of neurons in the near vicinity of that electrode. And then finally, you can actually measure similar types of field potentials that sort of aggregate and sum up neural activity if you put an electrode on the surface of the brain. We call that electrocorticography. And so originally people have really always thought about, so all of these signals have been used to control brain-machine interfaces successfully. We've also used EEG, which is a similar concept of these field potentials, but is actually measured from the surface of the scalp. And so all of those signals have been used to varying degrees of success for controlling brain-machine interfaces, but it's not necessarily clear which is the best choice of signal. And it's a large parameter space, but just thinking about some these sort of two signals in contrast, spiking activity is sort of often thought to be the obvious choice by many people because we know that it's very closely correlated with behavior. If you give me just a couple of neurons, I can build a decoder that gives you very accurate predictions of arm movements. The longevity of these signals is relatively poor. There's a lot of issues with the biological interface around these electrodes where you gradually lose the recording of action potentials. Also, there's a lot of relative movement, and so the specific neuron that you might be recording can change over days. So again, this measurement noise becomes a factor that might influence long-term learning. On the flip side, ECOG generally speaking, is thought of as kind of a low information signal. It has relatively poorly understood relationships to behavior. You can decode movements from it, but it's definitely less robust and less informative than spiking activity. The flip side is that the ECOG signals themselves, because you're summing from larger pools of neurons, inherently have lower measurement noise, and there's also potentially some biomechanical and biological interface benefits from electrocorticography. And so from a learning perspective, potentially these signals are going to be more stable, which could improve performance. And so again, from a decoding perspective, the choice between these two is pretty obvious, but if you start factoring in kind of issues of measurement noise and stability of the signals and how that impacts learning, it's not necessarily very clear. And in fact, I've also done some work where I looked at interfaces controlled with different signals and saw that the neural strategies, the activity in the brain when subjects are controlling interfaces with different types of signals seems to actually influence their control strategy. And so this is not really well understood, and it suggests that these choices are not necessarily inherently clear. And so rather than thinking about how much information we can get out of a signal, I want to start exploring which signal is actually easiest to learn to control and why. And that might give us some insights into some of these issues like BCI illiteracy, where we want to optimize kind of the learnability of these interfaces and the stability of them longer term. So one sort of project that we're planning on pursuing in the lab is doing sort of direct comparison of interfaces controlled with these different signals. So for example, you can think about having just a really simple brain-machine interface where you have a cursor moving in 2 dimensions, x and y, and you have 2 neurons that are controlling the cursor's x and y dimensions. You could alternately now have the same interface, but now controlled with ECOG signals, where you pick a certain property of the neural signal, in this case the power in a high frequency band, and use that to now control the cursor. And so this requires, to really do this comparison properly, you have to do a direct comparison. Sorry, just trying to keep track of time. A direct comparison where you have the same subject, the same location within the brain. And so really this requires being able to do these measurements of both ECOG and spiking activity simultaneously. And so the goal here would be that you could do the proper test to really see which of these 2 control signals gives you the best performance, both in terms of learning rate and then the final performance that you can achieve with those devices. And so, as I mentioned, this requires being able to simultaneously record these 2 different types of signals, which it turns out is a slightly technologically challenging, And so something that I worked on a lot in my postdoc is sort of a new approach to neural implants that would allow us to make all of these different simultaneous measurements. And so the approach that we did is, rather than having dedicated hardware that's for making a particular type of measurement, we had an implant into the brain that you can think of as sort of an access port that is agnostic to the type of hardware required to make the measurement, and then you can flexibly change the hardware out depending on what measurement you want to make. So what we've done is this access port, so to speak, is a minimal implant onto the skull, and then an artificial dura where we remove the dura that covers the brain and replace it with an artificial one. And then we can actually and so this is what that looks like, where now we've exposed the surface of a monkey's cortex. And the nice thing about this artificial dura approach is now we can actually instrument the surface. So we can put an ECOG array into the artificial dura so that we can now make measurements from the surface. And changing that out, going from a regular dura to an instrumented dura, is a non-surgical procedure that's very easy and reversible. And then we have this minimal chamber design that sort of allows us to flexibly stack up adapters. I like to think of it as we've implanted sort of the equivalent of the green Lego board that you then build your Legos on top of. Is the general idea of it. And so now we can stack up our Legos depending on what types of measurements we want to make. And so this system allowed us to do this multi-scale electrophysiology where we can, it's a little hard to see, but there are a bunch of little small dots on here. There's 244 of them that reflect ECOG contacts that we're measuring, and then there are these bigger holes that reflect where we can have penetrating electrodes go through. So now we can measure spiking activity on penetrating electrodes while also measuring activity from the surface. And this technique, because we're using artificial duras, is also compatible with optical techniques. And so we're very interested in starting to think about how to spatially register all of these different measurements that we're making together. And we want to do that using the vasculature. So we can take some very nice wide field images of the brain where we get fine, detailed resolution of the vessels. And we can even do that, through the holes in our arrays where for the ECOG, so that we can precisely locate where our electrodes are on the brain relative to each other. All right, so I think that gives us a good amount of time for questions. But to wrap up, the big picture goal of my research is to sort of explore learning in brain-machine interfaces and how we can exploit it to improve performance. And so a big part of that, which I focused on today, is this idea of revisiting all of the design choices that we make as we build a brain-machine interface, given that we know the brain is going to be learning. And so some of those approaches are adaptive decoding, co-adaptation, also thinking about how we design the loop itself, and then how we want to choose the signals, all with the goal of thinking about learning and control rather than decoding. And then finally, we're sort of pushing on some of these new ways to make measurements to study the brain so that we can get new insights into how the brain is learning to control these devices to move towards these design principle goal. All right, so thank you very much for your attention. And all of this work is sort of a mix of my PhD and postdoc. So the co-adaptation and loop manipulations were all done with Jose Carmina. The multi-scale measurement system was done at NYU, working with Bijan Pesaran. Here's some of my funding. And then finally, the lab is kind of just getting up and going. And so we've got openings both for all levels of research interest. So if you're interested, please get in touch.
So we have time for a few questions.
Yeah.
I appreciate that you're willing to look at both neural spikes and ECOG recordings in order to do whatever it is you need to do for whatever problem you're trying to solve, and that you want to figure out which one's better. But I'm curious, do you have any intuition going into it, which one you think is going to win out?
Yeah, that's a great question. So I think when I think about it from, there's several different ways that you can think about the problem that you're asking the brain to solve if you think about what you're asking the brain to learn to do. So if you think about just learning to modulate an ECOG signal versus learning to control the firing of an individual spike. The ECOG signal reflects activity summed up over, let's say, for the sake of argument, 100 neurons. And so learning to, I think about it in the context of redundancy. So if you have just one neuron that you have to get to fire, in some sense that's great because we don't care what all the other billions of neurons in the brain are doing. They can do whatever you want in order to get that neuron to fire. But you're also asking the user to figure out which one within a billion neurons is the one controlling the cursor. And so that to me actually seems like potentially a very tricky problem of credit assignment, right? The ECOG, on the other hand, I guess there's sort of two ways to think about it, right? Either it's redundancy with respect to the control redundancy of, you can have versus the sort of versus the issue of the control, the search space. And so it's actually unclear to me which one's going to win out between the two because they sort of are perfectly opposed. And so that's part of why I think it's very interesting. When I flesh it out and think most about redundancy, I think ECOG potentially actually could win, though.
Thank you.
Yeah. Yeah.
So in terms of BCI literacy, I was curious, the primate subjects that you had, how much variation did you see among individuals?
Yeah, that's a great question. So in the primates, so BCI illiteracy specifically is a term that comes from the EEG literature, and that refers to humans. So it hasn't been specifically documented in primates. That said, there's absolutely variability across subjects in terms of monkeys and their proficiency in doing these basic tasks. I don't know that I have a great quantification for you. And one of the interesting things, and part of why I think a lot about BCI literacy, is that the non-invasive community has just seen a much larger pool of people, right? So they have, hundreds, thousands of people have tried to control EEG brain-machine interfaces, but these invasive brain-machine interfaces have been used in a really small number of people in clinical trials, and then a very small number of monkeys in a research setting. And so potentially the BCI illiteracy effect might be there, but it's being masked by the fact that we don't work with that many individuals.
I have kind of a follow-up to that.
Yeah.
As you move from non-human primates to human subjects, to what extent do you think that learning is explainable from one human subject to another?
So trying to think about whether everyone learns the same way, or
Right, and if one human patient could describe to another how to execute a control strategy.
Right. Yeah, that's really interesting. So I think that the current clinical trials, from my understanding, every patient seems to initially start with a very different strategy, which I think depends very much on where the implants have been made and other factors, and there's sort of some tailoring and customization. In terms of whether, so current data suggests that there might be some variability there. In terms of the broader mechanisms, though, I think there will be a lot of similarities when you start drilling down into, in particular, given that they made this mistake, how did they incorporate that mistake to update, their control models? I think a lot of those principles will be the same. It's just the question of the starting point for each person. And I definitely think that's going to be different. Yeah.
Fantastic talk, Amy. In the middle study where you looked at the update rates, maybe I got the details wrong, but your fast update was once every millisecond and the slow was once every 100 milliseconds?
Yeah.
So the fast update rate's on the order of an individual spike, right? The time the neuron takes a spike is about a millisecond. Mm-hmm. So that would imply that the individual timing of the spikes are either important or informative. Whereas if you look at like a Mike Shadlin interpretation that it's only average firing rate that matters, you'd want to at least average over 10 or 20 milliseconds. So I wonder, do you think there's a sweet spot in the middle, or do you think you've kind of hit the optimum with the 1 and the 100 milliseconds?
Right. Yeah, that's a great question. So I definitely think there's some interesting follow-up studies to sort of more fully sweep the space of rates, because we've kind of only tested 2 extremes. And I don't know for sure. I think that the one other thing in that study that I didn't talk about is that another key difference there was using this point process filter, which is sort of a different encoding model of which kind of gets at this issue of like spike timing versus rate. And we were also able to show that leaving rates all the same, if you compare a Kalman filter, which is sort of a rate-based model, to the Poisson model, you actually also are getting an improvement from that as well. Yep.
Do you think there's potential possibly to include both spike and ECOG data into the same system to benefit from both?
Mm-hmm. Yeah, that's a great question. So I definitely think that that's an interesting possibility. And actually, Miriam Shinichi, who I've collaborated with quite a bit, is thinking a lot about developing sort of multi-scale decoders. There's some interesting mathematical challenges in there because spiking activity, is this Poisson process, whereas the field potentials are continuous signals. And so the decoding frameworks are slightly different, and the timescales of the decoders are different. But I definitely think it's potentially feasible, and a lot of the data suggests that the 2 signals actually have slightly different information about the neural processing that's happening. So integrating them together might be a very interesting direction. Yeah.
I wonder, I mean, this is great work, and I wonder if what you learn on how to design decoders for these kinds of interfaces, if you look forward with humans and non-invasive, for example, EEG-based, do you think that what you learn in terms of the coding algorithms could help inform how you would decode non-invasive interfaces?
Yeah, that's a great question. So I think that a lot of the sort of bigger picture issues of things like the timescales for adaptive decoding and the rates of control and other things like that, I think will be very similar between non-invasive and invasive because they're sort of more related to control properties of how people are controlling the devices, and then also the timescales of learning. And so I think a lot of that is somewhat independent of the measurement that you're making. And so I'm pretty hopeful that some of this would translate. That said, obviously, one of the embedded themes in the talk is that, changing one element has, propagating effects. And so it will be interesting to see how that bears out.
Yeah, last question.
Thanks for the overview. I was wondering, as to your intuition as to how one might take the two-dimensional control strategies into, the 27 DOF sort of 3D world. Do you have any beginning strategies as to how to approach that problem?
Right. So I have some initial ideas, a lot of them focusing on first trying to understand how people even control things in high dimensions. So most motor learning studies have always been studied in the context of reaching and pointing, so people just making simple two-dimensional movements. We actually don't really understand very much about how, for instance, do I learn to get my hand to a cup first and then figure out how to grab it? Or is the shaping of my hand learned at the same time as the transport of my hand, and things like that are sort of really unclear. And so I'm thinking a lot about some interesting sort of interface design tasks where we're trying to start to tease apart some of those questions of, are you learning degrees of freedom independently of each other, or are they kind of tied together in some, for instance, are the learning rates all the same? So that's sort of one initial strategy. In terms of adaptive decoding, the main issue there that you have to tackle is sort of the issue that these spaces now are redundant, right? And so there's lots of different possible ways that I can get my hand to do the same particular task. And so because of the high control space. And so there you can start thinking about optimal feedback control strategies and other things to make simplifying assumptions to deal with the effector redundancy.
Great. Well, thank you very much, Amy. It's great to have you here, and let's do a big round of applause.